So my name is Jorge Blay, and I work on numerous projects, one of which is amber and plant exudates. So plant exudates are a set of generally organic chemicals that plants ooze, generally out of their bodies. There are three major kinds of plant exudates. First one, resins. Fossilized resin is amber. Second, phenolics. Third kind, gums. As far as I am aware, neither phenolics nor gums have survived deep time. Now, the original goal of this project was and still is to have a universal library of plant exudates and ambers of the world using nuclear magnetic resonance spectroscopy. And we hope that some of these will be especially useful for the identification of unknown materials as well as those made by humans in the past. Plant exudates and exudates in general are oozed by all cells. In the case of plants, we know that all plants ooze these materials. However, because of the logistics of collecting them, we have concentrated from or on ferns towards the flowering plants. These materials are generally oozed by the above ground portion of the plant called shoot, but they are also oozed by the below portion of the plant or root. When I go to the field, which generally is botanical gardens where the plants are labeled, that's very nice. I'm just looking for the glistening stuff, you know, the shiny stuff when the light reflects on them or on areas of the plant where there is a cut. That's the easy way to collect them. Plant exudates are the ultimate result of the process of photosynthesis. The technique that we use, which is not the only possible technique that can be used, is nuclear magnetic resonance spectroscopy. Now, this is the absolutely key slide of this presentation. These are my two colleagues of so many years, of Joe Lambert and Yuan Wu. Depending where the peak is located along the horizontal axis, that's the basic module of exudate that we have. And when I say module, the implication is that we do not have, contrary to mass spectroscopy, we do not have chemical composition. We just have modules, basic building blocks, kind of like a Lego toy. So the basic reasoning is that I collect a sample, I get to have the fun part of this, send it to Joe Lambert, out come the analysis. After we do this many times, one good day we have a question. What is or was the botanical origin of this unknown sample? I just send the sample out, and out comes the analysis, and very simply, visually, we compare one to the other. And hopefully, we have the correct answer. Now, we have been working on this for very many years, and I will just now highlight a few of the major finds. You may remember that I said that there are three basic kinds of plant exudates: resins, gums, and phenolics. If one is a plant oozing a gum, we have not been able to do much in terms of classification. Yet, if one is a resin or a phenolic, we can do more with them. So here is an example of how, within a single plant family, the legume family, we have plants that ooze resins. We know that because there are lots of peaks on the right side of the chart, or others that have gums. We know that because they have these a peak somewhere between 60 and about 90 or so. Others ooze what we have quite recently called phenolics. They have peaks on several on several regions of the horizontal axis, and please note, very little on the right side of the spectrum. Now, what else have we done? We have actually discovered new NMR groups of phenolic exudates, as shown here. We have decided to call those xanthons. Or this very, very common in the Caribbean, Wayax, or balsam. We have been recently studying circum-Caribbean ambers and plant exudates, and confirmed the find that they are all within the same resin category. And we have used that information to confirm that pre-Columbian Caribbean inhabitants were making artifacts, maybe for the ears, using these types of materials. Also, there are, a gazillion materials that look like amber, and just to give one example, these we have found out to be just petrified woody plants. Thank you very much. The question is whether we are using this data to discover the origin of Dominican amber, and that has been clearly established. It's a legume from the genus Hymanea. Now, what's to me more interesting is that since every Puerto Rican, I am from Puerto Rico, it's a baby, we are taught that pre-Columbian inhabitants of Puerto Rico, the so-called Taínos, came island hopping, right? And we have pretty strong evidence that there is an alternative route, which is, directly from what's called the Isthmian region, that is somewhere between Panama or modern-day Panama and Costa Rica. Just, the guys and gals got into a big boat and went all the way directly through the currents. Some archaeological evidence actually support that, and whenever someone finds amber in Puerto Rico, which is, as far as I know, very rare, we at this museum have one of the 2 known samples, I'm always looking for additional evidence that will support that. Thank you.
Jorge, why plant ooze?
I have to remember the person who gave me this idea, which is my mom. She pointed out that in front of our home there was 3 oozing these materials, and yes, I know, but I think that I speak too loud. Can you all hear me okay? Yeah.
Webcast.
Very well. Okay, I will try to modulate, like a good singer. So I collected the samples, baked them, and asked myself the question, could someone be faking amber? So that's how I got into this, being very concerned that very first paper describing a new species of scorpion from the Dominican Republic, which I did with someone else many years ago, by golly, could someone have fooled me? And now we know that there are quite a number of fakes out there. And as usual, what originates as a little project has ballooned into this worldwide search for plant exudates and chemical signatures, et cetera, et cetera. And that's just one of my very many projects. First of all, the only sample I have ever purchased was dried maple syrup at a beautiful botanical garden in central eastern Ohio. As I failed in every single bucket, to find anything. I thought it would be easy, but I failed. So I went to the gift shop and got my sample. I think a part of the answer is that at least here in the USA, there has been a tremendous, a tremendous weeding out of the botanical sources of food for either ornamental purposes or feeding. There is a great piece of work by a guy who died all too early. I think he was from Yale University, describing the horticultural massacring that took place in the early part of the 20th century in this country, whereby, just like in Puerto Rico, just a relatively speaking handful of plants are used for ornamental purposes. So I suppose that a similar process, a similar weeding out, not because of any malice, but probably because of ignorance and getting used to the same thing, the same has probably happened with maple syrup. I can now think, for example, of agave syrup, which we can, fortunately get in the supermarkets, etc. Just by trying to remember what, the tremendous abundance of genera that actually produce gums all over the planet, your question makes me wonder, are there other societies elsewhere that use, other gums for, sweetening stuff? I just don't know. Someone very recently, as recently as last week, asked me, Jorge, I want to produce a sweetened vinegar. What chips of wood should I use? And I answered, to him, I'm going to send you my paper on legume exudates. Just look at the little pink boxes. That means gum, producing plants, and maybe that would be part of your answer.